Hello everyone, my name is Ruku Tokamo from Childline Kohima. We are so grateful to the Department of School Education, Government of Nagaland for giving us a slot to introduce Childline to you through this medium. What or who is Childline? Childline 1098 is a national toll-free helpline number for children in distress and in need of care and protection, which operates 24/7. It is a project under the Ministry of Women and Child Development, Government of India. Childline is currently operating in 570 cities all across India and presently operating in 5 districts in Nagaland, that is Kohima, Dimapur, Mokokchung, Peren and Kiferi. Who can call 1098? A child or any concerned adult can call 1098 anytime for children in distress and in need of care and protection. Childline do not reveal the identity of the person calling 1098. It is always kept confidential. What are the categories of cases supported by Childline? There are the missing children, the runaway children, abused children, children seeking medical assistance, child labor, street children, and all kinds of children in need of care and protection. What are some of the services extended to children by Childline? We respond to phone call 24/7, rescue of a child if need arise, nutrition and emotional support and guidance through counseling, referral services such as medical assistance, temporary shelter, education sponsorship, restoration and repatriation of a child to their families in the best interest of the child. How do we create awareness? We conduct awareness programs in college, schools, churches, colonies, outreach programs through one-to-one interaction, small group meetings, and distribution of our Childline IEC materials. We also create awareness on the existence of Childline and its services and the different laws of our country protecting our children. We also create awareness on the cases prevalent in our city. Childline does not work alone or in isolation. We work along with various line government departments and agencies like the district administration, the police department, the legal department, education department, medical department, labor department, the social welfare department, state child protection society, district child protection unit, the child welfare committee, our Childline partners in different parts of the country, and all the NGOs working for children. In conclusion, we want to send a message across to everyone that even as the entire world and our nation is battling against COVID-19 pandemic, Childline is still there to reach out to any child in distress and in need of care and protection. We also take this opportunity to make an appeal to each and everyone to please call 1098 whenever you see any child in distress and in need of care and protection. Let us all join hands to make our city a safe place for our children to live in. Thank you, God bless, and stay safe.
Hello everyone, my name is Monami from Rajashkri Government High Secondary School. I'm going to be your zoology teacher during this lockdown, and we're going to start with cell division that is found in chapter 10, remember that. I'm going to start with an easy chapter. This chapter is something which you have known since class 9 and 10, okay? So the first thing that comes to your mind when you talk about cell is that cell is the basic structural unit of the living body, right? So when you talk about cell, like in case you fall down, okay, and then you scratch yourself, after a certain amount of time it starts healing itself, right? So how does it heal itself? It heals itself by the process of cell division. Remember that. Your cell does not keep on dividing always. Remember that. Why? Because after it reaches a certain stage, it will stop dividing unless it is initiated, okay? Unless there's a reason for them to divide. That reason maybe because of cut in your body, or maybe because there is a division of your the sperm cells and the egg in case of female. So there are different reasons. So when you talk about cell division, we're going to focus on cell cycle. Remember the cell theory, right? It was in 9 and 10, which was given by Schliedian and Schwann, which talks about cell. Now we're going to focus on the cell cycle. It is a complete round type of cycle, okay? It is basically composed of 1, 2, and 3 stages. Remember, the first stage is the interphase stage. This interphase stage is also called the resting stage. Remember that, okay? The second stage is called the M phase. This M phase can be mitosis or meiosis. You know what is mitosis and meiosis, right? What do you mean by mitosis and meiosis? Mitosis is also called equational division. Why is it called equational division? Because the chromosomes, after division also, the chromosome number remains the same. That's why it's equal. Remember that. In case of meiosis, it is called reductional division. Why? Because the number of chromosomes becomes half, okay? Like in case of sperm and ova in female, right? In the reproductive organs. That comes under meiosis. So how do you define cell cycle? Cell cycle can be defined as the sequence of events, that means the different stages by which a cell duplicates, okay, its genome. What do you mean by genome? That means your DNA composition. Synthesize the other constituents of cells. What do you mean by other constituents of cells? It may be your cytoplasm, it may be your ribosomes, that is the organelles, okay, and eventually divide into 2 daughter cells. What is the main aim of cell cycle? To divide into 2. Remember that, okay? The cell cycle is of 2 types basically. When you talk about the M phase, that is mitosis and meiosis. So there are 3 stages in which there is the interphase stage. In this case of interphase stage, there is the G1S and G2 phase. Remember that. Why are there G1S and G2 phase? These phases are there in order to help the cell grow in number. That means the quantity becomes bigger and bigger, okay? The cell cytoplasm becomes bigger and bigger. That means they become ready. They get ready to divide itself. And then the chromosomes also become bigger in number, okay? But the quantity becomes bigger, but they do not increase the chromosome number. Remember that. So the interphase stage is composed of G1 phase in which the enzymes and substrates are made. Why? Because during every cell division you need enzymes to break down. That's the reason why here there's lots of production of enzymes. After that, what will happen is that it comes to the S phase where the DNA, that means your gene, starts replicating, but the number does not increase, okay? Remember that. It just becomes bigger, replicates. The DNA replicates in S phase, and then in the next phase, that is the G2 phase, what will happen is that inside your cell, you know that there is the cytoplasm as well as the nucleus, right? That is the genetic composition. Besides this, there are different type of structure inside the cell, okay, which will help in cell division. So in this case, the specific structure during cell division is the spindle fiber. It is a fiber-like structure which will help during cell division. That one gets manufactured in the G2 phase. Besides that, energy gets stored. What do you mean by that? You need lots of energy to break down. You need lots of energy to multiply and produce, right? So these are the stages where the cells rest, but they start multiplying and replicating. Remember that. That is the interphase stage. The next one is the Nfa stage. In case we're going to specifically focus on mitosis today. Next class, we're going to talk about meiosis. In case of M phase of mitosis, they are again divided into prophase, metaphase, anaphase, and telophase. Remember that. That means 4 stages. After this M phase comes cytokinesis. This cytokinesis, the word cyto itself indicates that it is talking about the cytoplasm of the cell. Remember that. That means there is division of the cytoplasm in this stage. Now comes another one structure you see here, right? G0 stage. That is the rest stage. What do you mean by this? It means that a cell will continuously stay in a state of rest unless it is compelled, unless it is instigated to develop or to divide. Okay, so a cell, like in case of your skin also, after it covers your whole body, your skin does not keep on multiplying and it does not become bigger and bigger, right? So that is because of the G0 stage. It knows that it should not replicate or multiply. Okay, so our cells stay at the state of rest unless it is compelled to what? Unless it is compelled to divide. Remember that. So in this class, in case of cytokinesis, that is the division of the cytoplasm. But in case of this M phase, it is also called the stage where karyokinesis happen. Karyokinesis. Karyokinesis happen. In this phase, it is called the karyokinesis because it involves the nuclear membrane. Remember that. The nucleus, okay, where there is division of the chromosome. You know that inside the nucleus, there is a genetic component called the chromosome, right? So the chromosomes have the DNA, right? So we are going to focus on divisions of the chromosomes. That is the DNA. That comes under karyokinesis. Remember that. So what are the various stages of mitosis? We're going to focus on the 4 stages, okay? The first stage of mitosis is the prophase stage. What is the characteristics of the prophase stage? When a cell comes under prophase stage of mitosis, what will happen is that inside the cell, slowly, you will see this structure, right? Slowly, the nuclear membrane starts disintegrating. That means slowly, it will start disappearing, okay? Besides that, there is a specific structure. It's a T-like structure, okay? That is called a centriole, C-E-N-T-R-I-O-L-E, centriole, okay? This centriole is the structure where the spindle fibers are present. The spindle fibers in G2 phase. The spindle fibers along with the centriole, what will happen is that they will start moving to the poles. What do you mean by poles? You already know in class 9 and 10 that when they move to the opposite side of the cell at the top, those are called the poles. Okay, so the centrioles will start moving to the poles. They have a specific function. I'll explain to you later. So in this case, the nuclear membrane disappears slowly in prophase stage, and at last the nucleoli, that means the nucleolus inside the nuclear membrane, okay, NuCleoli, inside the nuclear membrane, it will also slowly start disappearing. Besides this, you know that chromosomes are all twisted and they form a clump type of strands like in a normal cell, right? So when it forms a clump type of strands like in a normal cell, when prophase stage starts, slowly, the chromosomes will form such type of structure, this type of structure, ribbon type of structure. These ribbon type of structures are called chromatids. Remember that. C-H-R-O-M-A-T-I-D-S. Chromatids. This structure of the chromosome, that is the chromatids, will again further grow, divide their arms, okay? Divide their arms into 2 arms. These 2 arms are called sister chromatids. Remember that. So After it reaches the prophase stage, it will go to the next stage that is called the metaphase. In case of metaphase, what is the characteristic? What is the first thing that comes to your mind when you talk about metaphase? You will see that the centrioles have moved to the poles, right? After the centrioles have moved to the poles, the spindle fiber, I've already explained about spindle fiber. The spindle fiber will go and hold the sister chromatids. Okay, how do they hold it? By a suction type of structure, okay, that is found in the middle portion of the chromatid. That middle portion is called a kinetochore. Remember that, Kinetochore. That is a suction type of gap where the spindle fiber gets attached. Why? Why do you need the spindle fiber? Because it needs to hold the chromatids in the middle portion. Okay, in the middle portion, it needs to hold it steady. Remember that. When it's steady in the middle portion, these sister chromatids forms the equatorial plate. The word itself, equator, indicates the middle part, right? So they stay in the middle, attached to the spindle fiber through the kinetochores, and form the equatorial plate. Now, what will happen is that at the later stage of metaphase, slowly, this sister chromatid will be divided into two. That comes under the third stage. The third stage is called the Anaphase stage. Okay, you can't see properly, but Anaphase. The third stage is called the Anaphase stage. In this stage, what is the main characteristics? What will happen to the chromatid? When you talk about cell cycle, you are going to focus on the chromosomes. Okay, so the chromosomes has been changed into chromatids. Now these chromatids, what will happen in anaphase stage? They will get broken into two. Okay, after it gets broken into two, the spindle fiber will shorten. That means it will pull towards the opposite poles. That means the opposite direction where the centrioles are present. Remember that. These centrioles along with the spindle fiber will pull the sister chromatids towards the pole. That is pulled by spindle fibers, the sister chromatids. And then the centromere divides into two. So what will happen is that after it gets pulled to the pole, why does it get pulled? So that the cell will slowly divide into two, break into two. That's the reason why they're pulled to the poles, okay? So at late anaphase stage, at this late anaphase stage, chromatids gets changed back to chromosomes. That means what? This chromatid-like structure will start forming back into a ribbon-like structure. Okay, that is the same thing. It is composed of DNA only. So it will form into a ribbon-like structure that is called the chromosome. That particular structure is called the chromosome. Remember that. The next phase is the telophase stage. In this case of telophase stage, the most prominent thing is that the cytokinesis starts. That means the cytoplasm also starts dividing. That means a furrow is formed, F-U-R-R-O-W. That means it forms a dent-like structure where the cytoplasm also starts becoming two. So in case of telophase stage, the chromatids gets formed into chromosomes, which forms a network, okay, a thread-like network. That thread-like network is called the chromatin network. Remember that. Besides this, in this case of telophase stage, what do you see? Do you see this middle part where I have given the spot part? This indicates that slowly the nuclear membrane also starts coming out. So when you look at this diagram, you will see two nuclear membrane, right? So there will be two nuclear membrane that will start coming out and will start appearing. After this, what will happen is that slowly the nucleolus will also start appearing. And in due course of time, cytokinesis will completely happen. That means what? The cell will completely divide and then forms the result called two daughter cells. Remember that. This comes under telophase stage. Okay. So why is mitosis important? Why do you talk about mitosis, meiosis all the time? Why is it important? It is very important because it has identical gene composition. That means it saves the gene amount whether it divides or not. Okay, that means the characteristics is saved whether it gets divided or not. No matter how many times it gets divided, the chromosome number remains the same. Okay, if it's diploid, you know what is diploid, right? 2n number of chromosomes. It remains 2n number of chromosomes the whole time. Remember that. The second one is cell repair and replacement. I've already told you about the cell repair, right? Suppose you scratch yourself and then in due course of time you will see blood clotting, right? After the blood clots, slowly new cells come out. How does these new cells come out? It is possible because of mitosis, which gets initiated instead of staying in G0 stage. Remember that. So it gets replaced. So in this case of mitosis, it is different in case of plants and animals. Okay. The first thing that will come to your mind when you talk about plant cell is that when you look at the cell of animal cell and plant cell, I think it's found in your class 10 textbook, you will find a specific characteristic in plant cell called the cell wall. That is different. Okay, in case of animal cell, they just divide like that, with a furrow form and everything. Okay, but in case of plant cell, the cell wall has to be divided again differently. Remember that. So the cell division is a little bit different in case of the cytoplasm, in case of when you take into consideration the plant cell and the animal cell. Remember that. So this cell cycle, in case of this cell cycle, always remember our cells stay at the stage of G0 phase. That means a rest phase. Always remember that. Unless propelled or initiated. That means unless told to divide, okay, which comes under mitosis. Mitosis you'll find usually in case of cell replacement, cell repair, and then the same number of gene composition needs to be maintained. But in next class, we're going to focus on meiosis. Meiosis is also called reductional division. When you talk about meiosis, you should think about formation of sperm and ovum, where you know that sperm and ovum, they carry only half number of the chromosomes, right? So we're going to talk in next class about meiosis, which is basically focused on dividing the number of chromosomes into half. Okay, so that's all for today's class, and then see you next class when we'll start with meiosis.